So unfortunately, every time a conference is extremely well organized, such as this one, because Blanca's done an excellent job, they tend to ask you for your title and abstract long before you know what you want to talk about. So I've got no hesitation in changing that. So we'll talk about other stuff than I initially advertised, but we'll still talk about changing networks. So I want to start with something completely different. So whenever that was mentioned in Monty Python, they typically followed up a man with 3 buttocks. But here we start with a quote that was reasonably famous in some circles from 2007, where David Viniar, head of, at that stage, of financial security at Goldman Sachs, said, "We were seeing things of a 25 standard deviation move several days in a row." That is pretty much the most stupid statement about statistics that you can make. It's an astonishing statement in many ways, and this person managed to keep his job until last year, but there we go. So what is a 25 sigma move, a 25 standard deviation move? This is our normal distribution, and this is pretty much, here would be 3 standard deviations. This is 25 standard deviations. So David Viniar was looking at a system using a model, but clearly the model here was wrong. They based their predictions, their analysis, and their risk assessment on essentially what is a wrong model, a model which uses Gaussian noise to predict markets and so on. So we would like to keep this in mind, and the underlying theme of this talk is really how to deal with wrong models, how to make them better, and when do we believe we have a model that is acceptable. Briefly, just to give an overview of when you might want to sort of nod off or not, I will talk about changing networks as advertised. I will briefly talk about model selection, but at the level where it's essentially marriage advice rather than anything more formal. I will talk about hematopoiesis and then stability of stem cell dynamics and conclude. They have in common that here we think we have a model that is wrong, which we can make better. Here we have a set of models that we don't quite know how to make better, and we would like to choose which one is the best therapeutic model. And here is a problem that is related to this one here, where we can try to revive some 1970s theory from ecology and try to see how can we understand stem cell dynamics inside the hematopoietic stem cell niche. I will talk with the easiest bit here, the first bit of the talk, which has been extensively published, to talk about networks. So we are used to these network metaphors in biology. Here's the Berlin underground system, among one things, and here's a partial protein-protein interaction network in Candida glabrata, a pathogenic fungus, close relative of Saccharomyces cerevisiae. And we see them as a sort of graphic representation of complex data sets, complex processes, but we also have come to regard them largely as static. This one isn't static, so the tube doesn't, the underground doesn't work all day. It works different efficiencies, different times of day, and so on. And Candida glabrata's protein-protein interaction network will also change in response to physiological cues, to the environment, or to sort of internal triggers, which we don't easily capture in terms of these static network descriptions. So we have been interested in how can we bring this dynamicism into biological network models. And we take a reverse engineering point of view. That means we don't expect that we know how the system works. We would just like to understand some of the basic underlying mechanisms, which we ideally describe mathematically. There are sort of, on the statistical side, 3 generic frameworks, and we don't want to worry too much about them. They're Gaussian graphical models, Bayesian networks, and dynamic Bayesian networks. These are essentially directed networks. These are undirected networks. And they're not really Bayesian in a strict sense. They're just factorizations or probability distribution. But anyway, so we have come to use them to, for example, model co-expression or gene regulatory networks. But typically it was used in a way where we assume that the interactions are present or not, and we never have sort of any level of flexibility of turning interactions on or off in the simple framework. Typically, the inferred network structures represent only correlations rather than direct interactions. That is especially true for correlation networks or relevance networks that use mutual information. And we ignore a lot of interesting biology. So we ignore, for example, that activation might be required for certain transcription factors to be active and so on. And many of the factors that can also influence the interactions, especially external cues, are taken out of the description. And this is just one of the shortcomings that we would like to overcome in some way or form. And I'll just give a brief bird's eye view of how that can be done, and then go towards the other subjects. So in principle, we can model lots of things that we don't measure explicitly. And that is, we include them as hidden factors. The data does allow us to make some inferences about that. So if you take an extant set of DNA sequences, you can reconstruct the phylogeny because you have assumed there's an underlying structure. And we can do the same thing here. In the past, with a couple of postdocs of mine, we've looked at changepoint networks where we assume that the network changes at different time points, and we try to infer the changes in the network structure and when the network structure changes from time point series data. That is all fairly straightforward, but this was a method that in the end we didn't have that much faith in, especially in clinical settings where we don't have time course data, but patient, non-patient data. So there we were looking for more flexibility, and we want to put as few assumptions in as possible. So we want to take a parsimonious but not ill-informed approach, and we want to make it applicable to either Bayesian networks, Gaussian graphical models, and so on. I will not overly bore you with any of these sort of mathematical details. If you want to have the slides, feel free to email me. I'll be happy to send them to you. But I will, for most of the talk, assume that the network structure that we are modeling, whether it's a direct Bayesian network or Gaussian graphical model, that's fairly irrelevant. The underlying principles can be applied to both, and we have indeed applied them to both. What is more interesting is now how we look, for example, at time course data. That is easier to model than condition data, so patient, non-patient. And what we assume, we have some measurements, mRNA level, we can also include protein levels, and we have an underlying network that gives rise to that. So this blob here corresponds to a whole network structure, the gene regulator network, which is unknown. And as we go forward in time, the network structure can either change or stay the same. And for example, here we have cumulative changes and so on and on. And we try to map how the network evolves over time based on the measurements that we take at different time points. So far, so easy. The problem is we don't know how often the network changes. We don't know what the structure of the network is at each time point. So we have to somehow pull a trick out of the mathematical toolbox that we find useful. In particular, we would hate to make any assumption that there are 3 network structures over 20 time points when there could be 4 or 5 or 6. So we use a concept called Bayesian non-parametric inference, which does fancy things with concepts of infinity. In particular, it allows us not to specify any given number. And the best way of explaining what is meant by infinity here is a Chinese restaurant. And that's an analogy due to Aldous from 1983, where he says that in San Francisco, where he lives, but also in London, Chinese restaurants are essentially infinite objects. If you go into a Chinese restaurant in Chinatown, the waiter will always find another table to place you. And they're good examples. If you come to London, they're essentially places which you will never reach, they will never reach the capacity. So when a new patron comes into the restaurant, the waiter will guide this new customer to one of the existing tables, or he will open up another table for this person and start serving dishes at this table here. And then there is freedom to open more and more tables, and this is one way that gives rise to a process called the Dirichlet process. The Dirichlet process is a way of specifying prior distributions over other distributions. But essentially, it only does what a waiter in a London or San Francisco Chinese restaurant does. He opens up new tables, serves new dishes for new customers as and when they come in, and is effectively able to serve an infinite number of customers over time. So for us now, each time point is a table. Each time is a customer, and each network architecture is a table. And so we have to try to learn what dishes are served at different tables, and when do new time points get their own new table, or when are they assigned to existing tables. And that's what we're doing. And this is what is formally known as a hierarchical Dirichlet model. These are terms that are easily banded around. They're actually easy to understand, but they're frightful to people whose main obsession in life is not mathematics. And given that you're in a world-class biological institution, I assume that you don't have any interest in the deep mathematical or statistical points of this, so I just go through this at some level. So here we have a network where at different time points we assign it either to the orange, the mustardy or yellow network, and this purple network here. And the only extra assumption we have done is that over time, biological systems do not change wildly. That means if you go from one time point to the next time point, especially if it's very slow, you don't often see a dramatic change in the network architecture. Occasionally these things can happen, and that is what we want to learn. So we want to learn each of those networks inside here, and when the system changes from between time points. Each of those would be a data set, so your RNA-seq data, or ChIP-seq data, we can also include that. And each of those balls here corresponds to a full network architecture, our mathematical model of a gene regulatory network. That's what it would look like in mathematical terms. And this is, again, something which we've published extensively on and which we've sort of made almost user-friendly to use. But what is important here is that what we're interested in is this network architecture. Each of those dots is a full network, so the full gene regulatory network of these organisms at different time points. And the same process can also be applied to, for example, not time points, but patients versus controls, or even patients versus controls over time at different time points. What do we learn from this? And these are sort of the more mundane results that we get from this type of data here. This is a network where we just focused on a small subset. So computation is affordable to most people, this methodology. And I'll come back to that later perhaps. But what we have looked here is we have looked at the expression data for Drosophila melanogaster mid-gut development. It's all published data. And neither Tom Thorne, who did the work, nor myself know anything about Drosophila development. And we then try to see, are there any time points when the network changes? So there are no prior assumptions put in, apart from the fact that we allow for an infinite number of time points potentially. Obviously here only 11 with 12 time points. And what we find is that in the early stage, this type of network architecture prevails. At later times, the same set of genes is involved in many, many more interactions. So the second half, second 2/3 of these set of time points seems to be described by a much more vigorous set of genetic interactions or genetic regulatory interactions. That's one of the things which fall out if you then a posteriori talk to Drosophila developmental biologists at UCL or elsewhere, or you just look it up in published resources, then you find this actually makes sense. This is a point when we would expect some things to occur, certainly not down here. But when these things occur, it's just not a priori to us as ignorant people, not at all clear. We can also show for Arabidopsis thaliana, we have looked at a 24-hour data set, again it has been published, and we're trying to infer for genes which were chosen based on co-annotation. Do the network architectures change? And here we find one which operates at night and one during the daytime. So this is an artificial day-night cycle, and again you can find that the network architecture does change over time, and it does stick more or less to the previous time step apart from those situations when there is substantial statistical evidence that the network architecture changes. These are sort of the points where we have least idea. We have more idea about Candida glabrata osmotic stress response because the BBSSC fund us quite generously on this. And what we did here is we looked at metabolomic, but especially transcriptomic response. I'll talk only about the transcriptomic data in the context of osmotic stress response in Candida glabrata. And we find that the early response is regulated as associated with a different set of regulatory interactions than the later response. So the acute response and the adaptive response seem to be controlled slightly differently. Now, most of those things here, if you were to ask us to bet money, we would say most of those are co-regulation, not direct regulatory interactions. But occasionally we see something which pleases us a great deal. And this is a set of transcription factors which are embedded in here. And what we do find is if you look at the acute response, you find that ISD11 controls SMX3. After 30 minutes, ISD11 starts to also regulate BUD31, and SMX3 starts to control BUD31 as well. If we don't make the assumption that the network is allowed to change over time, we would not have gotten this interaction. And we would not have been able to find this interaction here either. We would only have to discover this interaction here. So by allowing the system to change over time, we can pick up some of these interactions that we would otherwise have missed. Now, the next point is, so this is in fact a sort of type feed-forward loop, where SMX3 seems to be, needs to be active before it can start to regulate BAT31. So we would not think, or we would not want to sell this as the gospel, the truth about regulation in Candida glabrata or any other organism, but we think that those are time-resolved hypotheses which you can test, because you can now go with RNAi or other mechanisms and try to inhibit, knock down, or somehow interfere with the network at given time points. And we predict, and in some other cases we have seen, that these effects will be different depending on what time point you interfere with the system. So in E. coli we've done this. So this is how we can, so we're quite happy that we can now deal with these, we can go from static networks to slightly less static networks on the way towards models that are more mechanistic, more dynamic in nature. What I'll now briefly talk about is the framework that we prefer, and here we've discussed other frameworks as well, but we are often faced with a scenario where we have some data, we have a set of different candidate hypotheses or models, and which we have here ranked by complexity, and we have different fits of the models to the data. And we always have to trade off the complexity of the model with the ability of the model to describe the data. Clearly this model here does a nice job of explaining the data, but it perfectly interpolates and overfits the data. This model here, nice parsimonious, but doesn't do a very good job, so typically somewhere in the middle lies the truth. And we and others are interested in formal ways of model ranking that allow us to choose these methods. Now, the particular framework that we prefer for aesthetic reasons as much as anything else is Bayesian model selection, where we assign a probability to each model to be the correct model given the data and given the competing models. This is something which is counter to what lots of people are taught at undergraduate statistics level. They're only taught that you can never accept, you can only reject hypotheses. But it is in fact not how we operate in day-to-day life. So when I decided to marry, I did not do so because I rejected 3.5 billion null hypotheses of women being the wrong one for me. So I attached instead a finite and large probability for my wife to be the one I want to spend the rest of my life with. You will see later on, if you pay attention, that this analogy will break down if I want to be a good husband. But anyway, so it is something in our day-to-day life, we try to assign probabilities of certain things to occur rather than rejecting it. And it turns out that all these classical hypothesis testing frameworks are approximations or simplifications of Bayesian model selection. So all these things that you come across, like a Kaiki information criteria and so on, they're approximations, sometimes quite crude, to this framework here. So that's what we're doing. So we now try to assess probabilities of models to be the true one. And we're looking at a completely different system, the Haematopoietic system. So every second of our adult life, we produce 2 million blood cells, about 10 to the 12 per day, 10 to the 11 to 10 to the 12. It's a very diverse system, a very complicated system, and we are interested in what controls the stability of such a system. And we take a population biological approach, partly because we've got a lovely collaborator, Cristina Lucelso, who produces unique data in many ways that I will show you in the next few slides. So we have here some of the cells that are inside the bone marrow, which produce our red blood cells, other blood cells as well. And we would like to see how are hematopoietic stem cells, or HSCs for short, maintained. There are a whole set of molecular factors which define this, but to good first approximation, these molecular factors are closely associated with certain cell types. So, for example, osteoblasts, macrophages, and so on, if you were to delete them, you will find that the hematopoietic stem cell niche in the bone marrow will change, and the dynamics of HSCs will change as well. So this one here is just about visible. Everything in red here is a hematopoietic stem cell inside a living mouse. These green cells here are osteoblasts, and everything in blue is bone matter. So osteoclasts, really. And we come now up with very simple models of HSC dynamics, which are hopelessly oversimplified, but try to capture some of the basic underlying mechanisms. So we have HSCs, which differentiate, the differentiated cells continue to differentiate and so on, and finally enter the bloodstream. So it's a very simple model. And with Christina's work, we can also inject leukemic stem cells, so cells harvested from leukemic mice. And here, for example, that's leukemic activity in yellow and vasculature in red. Again, inside a living mouse, but not a very happy mouse. But so we can look at what happens now if you have HSCs and LSCs, leukemic stem cells, inside the cell niche. The differentiation pattern, to the best of our knowledge, is slightly less sophisticated than in the healthy lineage. But this is actually very robust against changing this quite a lot. And we are now trying to take an ecological point of view about this, very much like we would look at foxes and rabbits and so on. And we compare different models. So there are different models have been proposed in literature for chronic myeloid leukemia, a disease which you can now treat with very, very high efficiency. These models differ sort of in various ways. In particular, they differ in the way how they model the niche. One model, our model, says that the niche contains many of the differentiated cells. This other model, M2, contains only the stem cells and the leukemic stem cells inside the niche, but all the progenitors and the differentiated cells are outside of the niche. So we have cells that compete for niche resources, cells that are reliant on the niche, and cells are completely unconstrained. So they don't have this sort of constraint or feedback that the niche can exert. And we now have some incomplete data. It's been published before, and we would like to see how does the niche, can we distinguish between these different models and how they treat the hematopoietic stem cell niche in light of such data? We can do so, we can sort of fit the different models, again, to the best of our ability to the data. We have data for remission and data for relapse. And all three models annoyingly fit the data perfectly. If you look at these models in detail, and this is just a horrible graph which doesn't come out quite nicely in this projector either, and also vibrates on the projector a little bit. But anyway, so what you do find out is that one model, which is completely devoid of any niche dynamics, would give rise to phenomenal levels of blood. If you had that type of chronic myeloid leukemia, you would be able to fill this lecture theater with your own blood in about 18 hours. So this is clearly something which is not realistic. Leukemia is not that bad. But anyway, so this other model does do a better job, but we can now compare the models based on the distributions that we obtain for parameters and models and so on, and try to see what distinguishes the model. And what we do find is that model A0, this model here, the mechanisms are shown here, but I just don't want to spend too much time on that. It separates quite clearly, so we do linear discriminant analysis, which tends to work perfectly in such of slightly complicated, poorly understood cases, much better than PCA, but as an aside. And we find that there are a couple of factors, dynamic rate constants of this population dynamic model here, that do distinguish and separate the cases that have remission from the cases that have relapse. For this other model, which only takes the LSCs and the HSCs into the niche, we don't find quite as good discrimination. So for example, here, these factors A2 and B2, which have to do with the proliferation of progenitor cells, they distinguish quite clearly the people who undergo remission from the patients who undergo relapse. So that's one thing which is quite neat. But in the end, despite the best statistical evidence, in light of the poor data that we have, we cannot decisively rule out either model A0 or M2. However, if model M2 is correct, then we would expect relapse to be much, much more frequent than under model A0, and the disease to be harder to control. So that now brings us towards the last part of the talk, which is the stuff I'm more interested right now in. We now have something where we have linked the population dynamics to some data, but in the end we come with another theoretical problem. Is stability of one, so this system is very unstable. A perturbation would drive you very quickly into a leukemic state or not, or into a relapse state. This model is more stable. So what makes these models stable? And this is work which was, again, comes back to the first slide, the stupid quote by David Viniar, because a few years ago the Bank of England decided that after the physicists have created the stock market crashes, now it's the time for biologists, especially ecologists, to see can we rescue anything. So they have looked at a couple of UK biologists, mostly from ecology and from systems biology, and asked what makes these networks stable. And so what's your basic guess? Which of these networks is more stable? Now, until 2008 or 2009, the UK banking sector would have said this network is much more stable. Fair enough, and that's where the biologists came in. So how do we define stability? And here these networks could be banks, but it could also be species in an ecosystem. By stable, we mean that the dynamical system, if you perturb it from its stationary state, goes back to the stationary state following a small interference. An unstable fixed point means that a small perturbation would drive the system away. This is just fairly banal mathematics, but we can make it less banal if we want to. But what essentially turns out, we just linearize a nonlinear system, and we look at the Jacobian of this dynamical system, we get some matrix. The interesting thing about the matrix, which in ecology is called the community matrix, is that if the eigenvalues, and that's the most mathematical concept we will require for the rest of the talk, if the eigenvalue of this matrix A have real part that is negative, then we are safe. Perturbations will always decrease in size and will go back to the stable fixed point. If they're greater than 0, then the system is unstable. I always talk about local stability, local instability, so local stability in this case. So, in 1972, Bob May showed in a paper that a system is almost certainly unstable if the interaction between two species scales such as this. So, loosely speaking, when the number of species in an ecosystem or the number of connections in an ecosystem increases, the system becomes less stable. He showed that by reference to a mathematical construct called random matrix theory, which has been powerful in lots of areas of the physical sciences, and increasingly also number theory, coding theory, and so on. The latest incarnation of this is called Gurko's circular law, and it shows that the eigenvalues of a large matrix lie inside a circle. Last year, Alesina and Tang generalized Bob May's results and showed that some random matrices, in particular those which correspond to predator-prey systems, tend to be more stable than random matrices which correspond to mutualistic or cooperative interactions. Something which is slightly counterintuitive, but it has been taken as gospel in ecology for a long time. So large, complex ecosystems will tend to be less stable than smaller ones. There's a couple of mathematics that we can do as well with this, and we now bring this. What we essentially doing is we look at different types of systems, and then we have a sort of fairly straightforward way of sampling lots of parameters, typically on the order of 1 million. For each of those parameters, we calculate the Jacobian, look at the eigenvalues of the Jacobian, try to see are they stable points reachable in a real biological system, are they stable, how stable are they, and so on. So that hides a lot of numerical effort, but it's not that exciting. What we're now looking at, and we can generalize this to banking systems and to ecosystems, always taking the domain expertise into account. So we're not saying that there's a simple sort of self-organized piece of thing that would work across all the different scientific disciplines. We always rely quite heavily on domain expertise. So we have different mathematical models of stem cell models. And we try to see, let's look at that in the context of random matrices. And Alesina and Tang have done that for a smaller number of species. So this circular law is remarkably robust. Even for sort of 3 or 4 species, you tend to observe something which doesn't look a million miles away from a circle. So I've looked at these models. This model in particular, here's the ODE equation. We find it has always 4 fixed points. And then we try to see how stable are the fixed points if you look at it as a random matrix. If you look at it as a random matrix but keep the structure of the interactions fixed, have just random parameters, or if we keep the structure of the matrix of these interactions fixed, but also now take into account the correlations between different types of parameters. So what we've done here in this column, we've sampled not the entries. So in random matrix theory, you sample the entries of this matrix are sampled randomly. We now sample parameters randomly, calculate the stationary states, and calculate the Jacobian around these stationary states, and so on. And we find the more structure you impose on these systems, the more stable such a system tends to become. Now, this is something which is reasonably different from what has been done here. So here at this level, everything was assumed to be more or less random. We are now, here they have started to impose some structure. They said it's random, but only predator-prey type interactions, which make certain impositions on the structure of the matrix that you have. In particular, signs of the different entries have to be opposite sides and so on. Also fairly gentle mathematics at this level. Now, what can we learn from this type of thing here? We can learn certain basic levels. So, for example, we find, like Bob May predicted in 1972, that stem cell systems which don't lead to lots of diversity tend to be more stable. This is pretty bad news for the haematopoietic system, because we know we have differentiation of this type here, or of this type potential. We don't quite know. But from this analysis here, it now emerges that if you take the random matrix approach that people have done in the past, you will always hopelessly underestimate the level of stability that real systems can exhibit. So this one would mean that no ecological system, no haematopoietic system could ever be stable. Or in less than 1/3% of cases, you would get stability. These are based on fairly sophisticated sampling approaches in high-dimensional spaces already. But so we trust these things quite a lot. What nobody has looked at in the past is how biologically plausible are these different fixed points as well. So this one, for example, we find is stable in half the cases. This one is never stable. This one is stable in about 1/4 of the cases. This one is 1/4 of the cases. So of the 4 fixed points, we have one which is, if you're in that fixed point, half the perturbations would drive it back to this. This one will never be stable. This one will be completely fair chance being stable, but not great. But much better than we would expect by chance. Now, this fixed point is 0. That means we have 0 stem cells, 0 progenitors, 0 differentiated cells, not biologically interesting. This fixed point is negative. It's not reachable, so we never have negative number of stem cells. Not even our mice. So they have 0 when we irradiate them long enough, but not below that. So that can never be reached. These ones here are biologically plausible, and it doesn't actually matter for the health of the organism whether it's this one or this one. So you could even switch between those 2 states and would all be fine at first level. We've done this for all these models. We've done this for a whole sort of other models, and here's the basic lessons that we can learn. So again, here is for a random matrix which has some sort of structural constraints which reflect the structure of a real system. And here is one which has also the correlations between the different parameters that make up these stationary states. What you do find, this number is always low, this number is always much, much larger than this one, and this number is always the largest number, give or take maybe a numerical error somewhere, a numerical sort of level of accuracy. For example, here there's numerically indistinguishable, but anyway, so this one should be always greater or equal than this one, and this one should always be massively larger than this one here. So we know from reality that we have large and complex systems that do appear, to the best of our knowledge, to be stable. Now, Bob May said large systems have a low probability of stability. And mathematically, this is absolutely correct if you take the random matrix ensemble as your sort of null model for how these things should operate. What we and others have found in ecology is that imposing structure upon such a system reconciles these views. And it's actually something which in the last paragraph of his 1972 paper, Bob May also mentions. And what we find interesting from a mathematical point of view, and that might be a fringe benefit in a sort of most biological audience, I fear, this is an incredibly rich field. Structured random matrices going beyond these simple simplifications that Eugene Wigner in the 1950s and then Girko and including some very, very eminent mathematicians have done. There are lots and lots of low-hanging fruit to be picked. It's quite an exciting area of research. So unusually, I'm ahead of time, which is slightly scary. But anyway, so that is fine. So I can procrastinate over the conclusions. So biological and natural, social, and technical systems are highly structured. So we know this. The underground, plane networks, linguistic networks, physical networks, the internet and so on, have an intrinsic structure. Now, in the first part of the talk, I suggested that looking at these structures being fixed in time is wrong, and we should encourage, or should have a more deeply ingrained perspective that says that these network architectures are in fact highly dynamic. They will change over physiological timescales, over developmental timescales, and across evolutionary timescales if you're in the realm of biology. Now, this should be taken into account, wherever possible, and it's actually surprisingly easy to do so statistically. We have found that the numerical effort is very, very modest. And I don't want to go back to the first slide, but if you were to look at the Candida glabrata data set, assuming a fixed network, it would take us something like 2 weeks on our computer to solve that. If you assume the network changes, we can do the same thing in something like 45 minutes. And why is that? In the first case, the model is so bad that you're trying to force something which is probably really dynamic into a very, very fixed type of structure. It's very much like if you try to fit your foot into a glove. That's an analogy which works better in German because they're called hand glove and glove. So a handschuh and is the German word for glove. But so it's a very, very poor fit to the real data that these models provide. These more time-varying or condition-dependent networks, they might not capture every aspect of the dynamicism, but they are a much easier thing to fit because they have much more of the realism that these real networks tend to exhibit. So that changed, I said the computational cost here is very modest. So this is just computationally very, very easy to learn. The other thing is the second part. So structured systems can be robust even if they're very large. And structure, in fact, induces stability in a way which we start to understand better by combining ideas from ecology with those from control engineering. And from the most biologically useful result is that in this hematopoietic stem cell problem that we're facing with Christina's data, we believe now fairly firmly, and we have some good mathematical and biological reasons to back this up, that the robustness is induced by population level feedback inside the niche. So that means that you have to have constraints on the number of cells that you have. You have to have ecological competition between different cell types inside these ecological niches, and that alone will drive the stability of these systems up quite considerably. The last lesson is one where my analogy from before falls flat. So just because you have a good model and a good fit of the model to the data doesn't mean that the model is necessarily a good one. So most of these statistical model frameworks don't require your model to be good, they only require your model to be better than the rest. And so that means you don't have to be very good to be the best if the rest is worse. And so that is something which makes the statistical life harder and is still an area of active research. And so in the end, the construction of models and their validation has to be based not just on the best statistics, and that's something we are fairly confident we can take care of, but also domain expertise. And that's something which is highly condition specific. So when we talk about banking networks, all we're trying to do is we're trying to focus on peculiarities of banks. You have different types of agents than other networks. Biological networks are much, much more straightforward and nicer to deal with than banking networks. Here are the people who funded most of this work and the people who've done the work, so foreshortened by the perspective here. But Adam McLean has done the work on the stem cell biology, and Paul Kirk has done the mathematical analysis of many of these stability problems. And Delphine Rolando, who I can't quite make out there, has done, she had to work with the bankers. She's the most patient person of the lot, but she wasn't there. So a bit early, but here I like to finish if there are any questions.